Hello, everyone, and welcome to today's The Scientist Symposium. I'm Tiffany Garbett, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our panel of speakers will present how synthetic biology accelerates the development of diagnostics, therapeutics, and the medicinal pipeline. We encourage you to send us your questions and comments at any point during the webinar, and the panel will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. I'd like to take this opportunity to thank our webinar sponsors, Beckmann Coulter Life Sciences, Isoplexis, Twist Bioscience, Acro Biosystems, and Codex DNA. Our sponsors have provided us with some helpful resources related to today's symposium, and you can find these in the handout section located on the right side of your screen. You can access and download these documents at any time during the symposium. And with that, let me introduce John Fuller with a brief message on behalf of our Diamond Level sponsor, Beckmann Coulter Life Sciences. John Fuller is a commercial product manager for Echo Drug Discovery at Beckmann Coulter Life Sciences. Prior to this role, he was a field application scientist for LabSite in the New England region and provided pre-sales and technical support for the Echo and Access automation platforms. He holds a BS in biology from the Virginia Military Institute, a PhD in cell biology and genetics from the University of North Texas Health Science Center, and completed a postdoctoral fellowship at Johns Hopkins University School of Medicine. Dr. Fuller?
Thank you, Tiffany. Is my audio okay?
Loud and clear.
Excellent. Today I'm going to give a high-level overview of our acoustic liquid handling technology with a focus on synthetic biology and a quick showcase for how some of our customers have repurposed their instrumentation for COVID workflows. But first, let's have a quick introduction to Beckmann Coulter Life Sciences and learn about the overall product offerings. Here at Beckmann Coulter, we make and support a broad portfolio of instruments and solutions for research use, from liquid handlers to cell counters to and centrifuges to flow cytometers. Along with that, we manufacture and support a range of high-quality reagents and consumables, which deliver high performance in a variety of assays from genomics to cell viability and flow cytometry. We also manufacture much of our own labware, including tips and microtiter plates. I'm going to now pivot to the Echo Acoustic Liquid Handler. In case you're unfamiliar, I'll provide a quick overview for the Echo technology, how it works. At the heart of the Echo is a transducer that generates acoustic energy, which focuses on the meniscus of the liquid in the source wells to dispense 2.5 or 25 nanoliter droplets, depending on the model, to the wells of a destination which is inverted above the source plate. The Echo uses proprietary technology called dynamic fluid analysis, which means that it can adjust the transfer parameters in real time for a variety of liquids. This means samples and reagents such as DNA, RNA, master mixes, and glycerol storage solutions, all at different concentrations, can all be transferred without any user calibration. All a user needs to do is select the right calibration from a short predefined list. By transferring from nanoliter to low microliter with high accuracy and precision, the Echo enables the reduction of reaction volumes and therefore reduces costs while maintaining high data quality. Paired with our revolutionary technology is friendly user interface software. Our Echo Cherry Pick and Plate Reformat software allows for both uploading CSV pick lists for assembly reactions or setting up variable amounts of reagents during a cell-free protein expression reaction setup, for example. Our Echo Plate Reformat allows for a very easy and straightforward drag and design sort of reaction setup, the type of a walk-up example. Now let's dive into some different synthetic biology applications that the Echo is used for in order to enable more efficient, faster, and cheaper workflows. In a typical gene assembly workflow, the Echo can help with very fast and low volume pooling of the fragments and adding master mixes, setting up gene assembly reactions. After the assembly and transformation, the Echo can easily array the transformed cells in the culture to get single colonies. The Echo has also been useful in optional QC steps, such as colony qPCR or NGS. When the echo is incorporated into these workflows, our customers see a reduction in the cost of the reactions and a simultaneous increase in the speed for such workflows. This paper is a great example of the echo being used with common gene assembly kits to miniaturize the reaction volumes and save on cost per reaction. This paper from the Chi Lab. They've shown that when miniaturizing the reaction volumes for either Gibson or Golden Gate assemblies, they see an increase in assembly efficiency while reducing the cost. For example, for Gibson assembly, going from a 20 microliter reaction down to 1 microliter, they got the most efficiency for the assemblies while simultaneously reducing the reaction costs from over $2.50 per reaction to just a few cents. The same was observed for Golden Gate chemistry as well, with a maximum efficiency of the reactions observed at 500 nanoliters. So far, we've talked about the cost savings of the echo through miniaturization of reaction volumes. But cost savings-based miniaturization is only part of the value from the echo. Just as impactful is the significant time savings. Based on this data from Amyris Biotechnologies and other customers, the echo has been proven to save about 15 hours in a typical workflow that would take 18 hours. So for an example, if an assembly would take 12 hours on a traditional tip-based liquid handler, it would only be 3 hours on the echo. And the pooling and normalization of the NGS QC step goes from 6 hours on a traditional liquid handler to only 12 minutes on the echo. This enables creating a lot more constructs, not only at a much lower price, but much faster. And this is why the echo has been adopted by a lot of our high throughput SynBio customers working in these applications. These high throughput DNA assembly and pooling workflows used by our SynBio Foundry customers have also found a purpose in repurposing their equipment and expertise in the COVID fight, such as exemplified by our customers at the London Biofoundry. In this particular example, the echo, in conjunction with other elements of their automation platform, was repurposed for automated miniaturized qPCR, as well as RT lamp-based reactions. Typically, when we talk about the echo, we say typically any source well to any destination well, but we don't even need a quote-unquote well. In this example, our customers use the echo to array bacteria clones onto SBS, SLAS compatible trays. When it comes to arraying colonies after the transformation, the echo can be a great enabling tool for high density and high throughput automated colony arraying onto media or agar trays. This method is much less error prone than traditional methods due to the nature of the non-contact dispensing. And just as a bright spot, or about 25,000 bright spots to highlight, we have in the bottom left a scientist from Jeff Buca's lab at NYU. They've used their echo to showcase their creative side, to create beautiful artwork by dispensing yeast encoding colored pigments onto the Omni tray or similar Petri dishes. These are 25,000 spot, 2.5 nanoliter arrays. You can see more amazing work like this at yeastart.org. To just drive home the use of the echo in arraying applications, let's now pivot to a great showcase for how arraying samples with the echo in a non-plate format is currently being used in COVID research. This publication, which highlights a great use of the echo in dispensing phage display clones that express immunogenic peptides. The customer used the echo to assemble NGS reactions to sequence phages displaying overlapped amino acid fragments that were immunoprecipitated from pandemic positive sera. And then they subsequently arrayed those clones at 25 nanoliters per spot to confirm the antigens. They stained the clones with positive sera, and then that was normalized to the T7 in the library with a duplex fluorescent stain. I, of course, can't do this study or the ones from the London Biofoundry or the Chi group the justice that the authors can, so would ask that you read their papers, but do want to highlight these methods for rapidly developing diagnostics research. Another highlight where the echo shines in synthetic biology workflows is the setup of cell-free transcription, translation, TXTL, or cell-free protein expression systems. These are versatile platforms for production of recombinant proteins within synthetic biology. The very fast, accurate, and precise low-volume transfers of reagents and DNA pieces from the echo allows for up to 10x faster reaction optimizations while lowering the reagent consumption and costs by at least threefold. I've highlighted examples where the echo adds specific value in SynBio workflows, and we can add even more value with direct integration of the echo in biomech. As shown in the picture here, we have an Echo 525 in a biomech. We can span the range of liquid handling from the nanoliter domain all the way to the milliliter. Faster, low cost, and high throughput sample processing are achieved by combining the speed and low volume accuracy and precision of the Echo acoustic technology with the flexibility of the Biomech I-Series workstations. In such platforms such as this, the gripper arm of the Biomech will hand over the plates from the Echo to the Biomech and the onboard devices and vice versa. At Beckmann Cultural Life Sciences, we offer full product solutions from the instruments to reagents and software, all with the same great engineering and application support from one company to make sure we are a supporting partner in your research journey and endeavors. And thank you.
Thank you for that presentation, Dr. Fuller. With that, let me introduce our first speaker, Anum Glasgow, who earned her PhD in bioengineering from the University of California, Berkeley, where she engineered a bacterial secretion system to export biopolymer proteins for materials applications. Presently, Glasgow is a postdoctoral scholar in the laboratory of Tanya Kortama at the University of California, San Francisco, UCSF. She received an NIH NIGMS K99 K00 postdoctoral fellowship, a UCSF IRACDA postdoctoral fellowship, and a UCSF Chancellor postdoctoral fellowship. Glasgow works to understand and rewire signal transduction in allosteric proteins. Over the last year, Glasgow, members of the Kortama and the James Wells Laboratories at UCSF, and other collaborators joined forces to design ACE2 receptor traps for SARS-CoV-2. Dr. Glasgow, please feel free to begin.
Hi, thanks for that extensive introduction. Super excited to be here today and to hear some cool talks about SARS-CoV-2 therapeutics and diagnostics. And today I'm going to tell you about a project that I worked on over the past year in collaboration with my lab mates and colleagues in the Wells Lab at UCSF, including Jeff Glasgow, who's also here. Our project was to build ACE2 receptor traps as therapeutic candidates to treat SARS-CoV-2 infections. I'll also tell you about some ongoing work in our labs to build other types of therapeutics for SARS-CoV-2 as well as a diagnostic. So a SARS-CoV-2 infection begins with an interaction between the virus and the human receptor protein ACE2 on the surface of cells. The SARS-CoV-2 virion is covered in spike proteins. This is this gorgeous trimer, which I'm showing here, with each protomer having a receptor binding domain, which is in pink. And this receptor binding domain has different conformations, an up conformation and a down conformation. And it can interact with the receptor protein ACE2 with a binding affinity of 10 to 20 nanomolar. Our idea was really simple, which was to build a receptor trap. When you make a receptor trap, it's a therapeutic candidate that works by outcompeting the interaction of this natural interaction between a human protein and the viral antigen. And so we set out to engineer a variant of the ACE2 extracellular domain that it binds to the spike protein more tightly than natural ACE2 can. So the idea here is that you trap the virus by saturating the outside of the virion with your receptor decoys or receptor traps, and this blocks cell entry of the virus and halts the infection. Previously, GSK held phase 2 clinical trials to study the safety of human recombinant wild-type ACE2 in subjects with acute respiratory distress syndrome and acute lung injury. And now, Apeiron Biologics is testing the same molecule, or a very similar one, as a treatment for COVID-19. And I think the trial is over and the results should be out soon. And so a major advantage in using a receptor trap for viral infections is that if the virus mutates, and can avoid neutralization by the receptor trap, its cell entry mechanism is likely to also be inhibited because it uses human ACE2 on cells as a point of entry. And so engineering a tighter binding ACE2 trap could enhance this effect, and that could reduce the necessary dosage to block the infection and reduce viral escape. We decided to use a combined computational and experimental strategy to re-engineer the extracellular domain of ACE2 to improve its binding affinity with the spike RBD to build our receptor trap. So the first thing we did was a computational alanine scan at this interface to find hotspot residues, and these are residues that contribute most strongly to this binding interaction. And we're most concerned with the hotspots on the ACE2 side of the interface because the spike does what it wants, but we can engineer ACE2 to bind the RBD more tightly. And so we identified what are these hotspots, these residues that dominate binding to the spike protein. And we focused on position 34. Now, the natural amino acid occurring at position 34 is a histidine. And we did saturation mutagenesis computationally at this position to find what impact mutating to other residues at this position could have. And we evaluated the impact of these mutations by several metrics, including the predicted summed pairwise interface energies in the background of each mutation. And we compared these to wild type. And so you can see that in this graph over here. Lower energies are better. And we found that a valine at position 34 is predicted to be the most energetically favorable solution, while an isoleucine at the same position disrupts all of the interactions in its vicinity. And so we decided to redesign this part of the interface in the context of both the valine and isoleucine mutations at position 34 to generate diverse solutions to this problem. We used a flexible backbone design algorithm to do this, holding constant at position 34 both the valine mutation as well as the isoleucine mutation. And we allowed residues on the spike side of the interface as well as the ACE2 side of the interface to change conformations and the positions of the atoms in the protein backbone and the protein side chains. And we also allowed some positions in the immediate vicinity of position 34 on the ACE2 side of the interface to change their amino acid identities. This generated some conservative solutions that included no additional mutations in the case of a valine mutation at position 34. But when we make this really disruptive mutation to an isoleucine, we have to have additional mutations to accommodate that disruption, leading to what we predict to be an equally energetically favorable solution, which I'm showing as computational design 2 over here. So we created a handful of these computationally designed receptor traps and tested their binding to the spike RBD in vitro using biolayer interferometry. And we tested these proteins as fusions to FC domains from human antibody, which has been shown in many therapeutic strategies to improve the serum half-life of the drug. So in our experiment, we were excited to see that these computationally designed traps worked to outcompete the natural interaction with ACE2 right out of computational design. We saw that both our computational design 1 and computational design 2 had improved binding affinities or lower KDs as compared to the wild type ACE2 RBD interaction. And then we built an additional computational design number 3, which had 2 additional modifications. The first was that we mutated N90 over here where there's a glycan to an alanine to avoid this glycosylation, which was shown in KK Chan et al., which at the time was on bioRxiv, to possibly improve the binding affinity of this interaction. And then the peptidase domain of ACE2, which we had been engineering, also has natural catalytic activity. And so we were interested in knocking this out by mutating some of these residues in the active site to avoid off-target effects from angiotensin cleavage. And so we made our computational design number 3 with these additional modifications in the background of the H34V mutation. And this also had an improved binding affinity as compared to the wild type ACE2 spike interaction. So we pulled these effective receptor traps with modestly improved binding affinities for the spike RBD in an error-prone PCR library so that we could use an evolution strategy to get even tighter binding receptor trap for the spike RBD. And we did affinity maturation using yeast display, and we applied 5 rounds of increasingly stringent selection to isolate clones that were receptor traps that could bind more tightly than their computationally designed parents. And so I'm showing one example of these type of data here. So here's a clone from our 4th sort, shown in olive. And you can see that it binds more tightly to the spike RBD than its computationally designed parent, which is shown in green. And you can actually, on the surface of yeast, measure the binding affinity, the monovalent interaction with the spike RBD. And we did this for several clones that we isolated from different rounds of our yeast display campaign. For our tightest binders from sort 5, we could observe a 170-fold improved higher affinity with binding affinity nearing 100 picomolar. And it turned out that not all of these mutants expressed as well in mammalian cells as Fc fusions as they do on the surface of yeast. And so we moved forward with a clone from our 3rd sort, which was pretty robust and which had an improved binding affinity and which we could purify with high yield. At the same time that this was ongoing, we were really interested in some results that KK Chan et al. Published on bioRxiv using a deep mutational scanning strategy. And so as an orthogonal approach, we built a small set of deep mutational scanning guided ACE2 receptor traps. KK Chan et al. Mutagenized 117 sites on ACE2 in the spike binding interface and used a mammalian display with FACS strategy to try to enrich for single mutations that improve binding to spike. And we were fascinated by this because a lot of the positions that they mutated also turned up in our yeast display campaign as having mutations that could improve the affinity of binding to the spike protein. But in addition, they discovered several mutations that were not at positions that we had mutated, not at alanine scanning hotspots, and not even in the interface, that also conferred an advantage and were enriched in their assay. And so we wanted to do another computational saturation mutagenesis at some of these positions that were in the interface, but not at alanine scanning hotspots. And we combined some of the energetically favorable mutations by interface energy calculations at these positions from computational saturation mutagenesis that were also enriched in this DMS experiment to build a few more DMS-guided designs. So these have mutations that are not at hotspot positions in the interface, but appear in both this DMS experiment from the Proco lab as well as in our computational saturation mutagenesis. And they're presumably serving some kind of stabilizing role here. So we built 4 such DMS-guided designs that combined these types of mutations with mutations that improve the binding affinity as we predicted computationally, and used the same binding assay on the surface of yeast to test their monovalent KDs for binding the spike RBD. And these are also pretty good, with up to 51-fold improved affinity over wild-type ACE2 interaction with the spike. So at this point, we've generated affinity enhancing mutations using 3 different strategies. First, a purely computational design approach, and then we affinity matured these to yield additional mutations through yeast display. And then finally, we used this orthogonal approach using these DMS data that yielded different solutions. We wanted to put all of these mutations in a good scaffold where we could stably express an ACE2 that could accommodate these mutations and could have additional advantage when we use them in a therapeutic context. So at this point, all of the engineering that we had done was in the peptidase domain of ACE2 and the spike interface, or nearby there. And so we saw another structure of ACE2, which included additional structural domains, including the collectrin domain, which I'm showing here in yellow. And we were pretty excited about this because it looks like the collectrin domain serves to stabilize the the additional extracellular domain of ACE2 as a dimer on the surface of the cell. So we already had included an FC domain, but we thought that including this collectrin domain could further stabilize our construct and allow for more avid interactions with the full-length spike, which of course has 3 RBDs, thereby improving the affinity that we can observe. And so to test this, we decided to build an additional construct which included the collectrin domain. And we tested the binding with and without the collectrin domain of ACE2 against the spike receptor binding domain, as well as the full-length spike, which includes all 3 receptor binding domains, hypothesizing that increasing the number of these interactions through the inclusion of the collectrin domain and the additional spike RBDs could result in a tighter binding interaction. And this is exactly what we saw. And so here on the left, I'm showing some BLI data, in vitro binding assay data, from ACE2 interaction with a spike RBD. When we add the collectrin domain in this middle plot, we can see an improvement of about 4 to 5-fold in the KD, in the binding affinity. And then all the way on the right here, where we include the collectrin domain and the additional RBDs, we see a more dramatic improvement in the off rate, which is so low that we can't even measure it using this assay. And so this helped to inform the scaffold that we wanted to use for ACE2 receptor traps. We included the receptor collectrin domain and the FC domain, and then an additional inactivation mutation. We decided to test these receptor traps using a pseudotyped lentiviral assay in-house. And so in this experiment, we use a lentivirus to infect ACE2 expressing human cells. And the lentivirus contains a gene encoding luciferase and includes a SARS-CoV-2 spike proteins in its coat. And when the cells are infected, they luminesce, which we can measure. And if we add an inhibitor like our ACE2 receptor trap, it should block the infection. And so we can determine an IC50 in an inhibitor titration experiment by monitoring the decrease in luminescence. So we started out with our pseudotype lentiviral assay by testing wild-type ACE2 monomer against the wild-type ACE2 FC. And this is a useful experiment because, as I mentioned before, the wild-type ACE2 protein is currently in clinical trials in Europe. And we wanted to see what is the effect of just adding this FC domain. And so we see a drop in the IC50 just from the forced dimerization through including the FC domain. When we test some of our designed ACE2 receptor traps, we see a more dramatic improvement in the IC50, with the best result coming from our sort3 mutant, the affinity matured computational design, which had an observed IC50 of 28 nanograms per mil. So we sent off some of our best receptor traps from our pseudotype lentiviral assays to our collaborators at the University of Alberta in the Tom Hobman labs. Tom Hobman and Daniel Limonta did these experiments. And they moved super fast to test our receptor traps in their authentic SARS-CoV-2 neutralization assay in BSL-3. So in their cell culture assay, they could get IC50 of 73 nanograms per mil from our best receptor trap, the sort3 mutant. And this is on par with neutralizing antibodies that have been isolated from the B cells of infected patients. And so we showed in this project a general approach for combining computational design with affinity maturation using a surface display to engineer improved receptor trap therapeutics. Some advantages of using receptor traps to treat viral infections is that they're quite robust to viral escape mutations. This is something that we're testing more rigorously now in collaborations with our collaborators. I'm not allowed to talk about this data yet, but what we're testing is how many rounds of passaging does it take to allow for viral escape using our receptor traps and other types of inhibitors. We also tested the binding of our receptor traps to other types of coronaviruses. On the top here, you can see binding data for COVID-19, SARS-CoV-2, but we also tested it against the original SARS RBD from SARS-CoV-1 and HCoV NL63 RBD, which is something that can cause a common cold, this binding interaction. And our receptor traps bind all of them. And so you could imagine improving on these to treat other kinds of viral infections. You don't need an infected population to build a receptor trap therapeutic. All you need is a structure. And so you could imagine stockpiling pre-designed and improved receptor traps for other types of pandemic viruses. And we imagine using these receptor traps possibly in combination with neutralizing antibodies that can bind epitopes outside the ACE2 interface, or with other spike binders, or with other therapeutics. And this is something that we are also exploring now. The project took about 2.5 months. It was really fast as a result of extreme teamwork in our labs, and it was really fun. And so to give you an idea about how this project went, we went into lockdown or I should say shelter-in-place orders in San Francisco in mid-March. And within a week, we had our first set of computationally designed receptor traps. So we quickly ordered the genes and cloned these and purified them, and did our first set of in vitro binding experiments to get some results within 2 weeks. And then the same week, we confirmed that wild-type ACE2 could display on the surface of yeast, which makes this a viable engineering strategy. So in the next 3 weeks, we finalized our yeast display library and started our affinity maturation campaign, and then cloned everything over the following week into our collectron-containing scaffold and got our first viral neutralization data from our collaborators in early May. And by the end of May, we were characterizing our tightest binders from yeast display and their neutralization efficacy in cell culture. Now we are working in mice for our best receptor traps in collaboration with Jay Cole's lab at Tulane to measure the efficacy and the biodistribution and the pharmacokinetics of our receptor traps in both the active and inactivated form of the traps. We've also got ongoing structural studies in collaboration with Soumya Govindaramesh in the Wells Lab and the QCRG Structural Biology Consortium at UCSF to determine whether the design proteins that we tested are what we designed them to be, and to confirm what is the mechanistic basis for these additional affinity-enhancing mutations that we found in yeast display. We're also continuing to engineer these receptor traps for preclinical development to make them more stable, and also exploring the possibility of using them as fusion proteins or in cocktails with other spike binders. One such spike binder has also been developed at UCSF in the Wells group and the QCRG. These are antibody fragments, VH domains, and Bracken et al. At the same time that we were building our ACE2 receptor traps, they built a VH library, which they then evolved using phage display to find binders that could bind in the ACE2 and other interfaces, and combined these in several interesting geometries and formats to test their neutralization against SARS-CoV-2, and did some beautiful structural work. And this could be like a possible additional binder that one could combine with our strategy. In addition to these follow-up studies that I mentioned, people are using our receptor traps as tools for building diagnostics and probing the biology of SARS-CoV-2 infections. And one such use of our traps was by our colleagues here at UCSF, who are working on developing a serology assay while our project was ongoing. And they used our ACE2 traps to quantitatively determine increased sensitivity in their assay with increased affinities of binders in their samples. So the way that this serology assay works is that in your patient sample, you might have antibodies, and Susanna Elledge and Xin Zhou worked to build protein sensors. So these are spike or nucleocapsid proteins that are transcriptionally fused to a split nanoluciferase. Split nanoluciferase has a large bit and a small bit component. These are both fused to the S or N protein, and when they bind to the antibody, they increase the local concentration of these split components, which allows for the complementation of the luciferase, and in the presence of the substrate, furamazine can do this reaction to result in luminescence that we can measure. And the way that their assay works is you have a human serum, you heat and activate it, and patient sample is then diluted in a plate. You add these protein sensors, whether they're S sensors or N sensors, wait a short amount of time, about 20 minutes, add the furamazine substrate, and then plate read. And this is much, much faster than a traditional ELISA serology assay, but works just as well. And our colleagues used this assay to test several cohorts in San Francisco area, as well as in New York, both inpatient and outpatient cohorts, and found that both the S sensors and the N sensors were sensitive and specific to detect patient antibodies in the samples, with the N sensors being actually even more sensitive than the S sensors. Some advantages of using this type of serology assay, in addition to how fast it is that it's quite cheap, it's accurate, it's quantitative, you don't need a lot of sample, works on whole blood, and now they're working on making this amenable to handheld detection and point-of-care diagnostics. And in our hopefully soon-to-be post-vaccine world, you can imagine using this type of assay for studying how variable human responses are to receiving the vaccine, how different antibodies are made in response to the vaccine, and whether they're resistant to emerging variants of SARS-CoV-2 with mutations in the RBD as well as the other domains of the spike protein. So if you're interested in this study, check it out on medRxiv. So this concludes my presentation. I would like to acknowledge all of the people that Jeff and I have had the great pleasure of working with at UCSF and elsewhere, including the University of Alberta, where our virology collaborators are, and our funding sources. Thanks a lot. Happy to take any questions.
Thank you, Dr. Glasgow. As a reminder to our audience, you may ask questions at any time using the Q&A tab. Our panel will have a chance to address these during our Q&A session. Our next speaker is David Baker, who is the director of the Institute of Protein Design, a Howard Hughes Medical Institute investigator, a professor of biochemistry, and an adjunct professor of genome sciences, bioengineering, chemical engineering, computer science, and physics at the University of Washington. His research group are world leaders in protein design and protein structure prediction. He received his PhD in biochemistry with Randy Sheckman at the University of California, Berkeley, and did postdoctoral work in biophysics with David Argard at UCSF. Baker is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. He received the Breakthrough Prize in Life Sciences, Irving Segal and Hans Neurath Awards from the Protein Society, the Overton Prize from the ISCB, the Feneman Prize from the Foresight Institute, the AAAS Newcomb Cleveland Prize, the Sackler Prize in Biophysics, and the Centenary Award from the Biochemical Society. He also received awards from the National Science Foundation, the Beckmann Foundation, and the Packard Foundation. Baker has published more than 500 research papers, been granted more than 100 patents, and co-founded 11 companies. 75 of his mentees have gone on to independent faculty positions. Dr. Baker? Dr. Baker, you'll just need to click on the microphone icon to turn on your audio. See? Can you hear us, Dr. Baker? Okay, so right above, there's the microphone tab. Perfect. Now we can hear you.
You can hear me. Okay, sorry about that.
Loud and clear. No problem. Please feel free to begin.
Well, yeah, it's very nice to be here, and I'm really lucky there was such a wonderful talk just before me, just now that you just heard on the ACE2 traps, and some of what I'm going to tell you about is going to echo some of the things that you just heard. So, which is good. So you'll get a double dose today. So I'm going to tell you about the design of proteins that the protein design we've done against coronavirus, And I'm going to, perhaps at the end, if I have time, say a little bit about sort of new approaches for protein design that we're using, we're developing using deep learning. But what I'm going to tell you about in the first part of the talk is all based on the idea that proteins fold to their lowest energy states. And so that means if you want to predict protein structure from sequence, you have to search through the different possible conformations of protein looking for its lowest energy state. And if you want to design a brand new protein, you have to find a sequence whose lowest energy state is that structure you want to make, say a coronavirus therapeutic. We've been developing, me and our colleagues have been developing this program called Rosetta over the years to do the protein structure prediction and protein design work. And one of my close colleagues is Tanya Kartemi. We just heard about Tanya on the previous slide. So what I'm going to talk about today is de novo protein design, where we're making up completely brand new protein sequences and structures. So rather than taking a naturally occurring protein and modifying it a little bit, we're building completely new proteins. And you'll see a lot of brand new proteins during my talk. And this is interesting because the sequence space is really, really big. There, 20 amino acids at each position. 100 residue protein would have 1.3 times 10 to the 130th conformations, and that's a really, really huge space. So it's far, far larger than what's been sampled during evolution. So there's a lot of room to make completely new things. Okay, so I'm going to tell you about our work on antivirals, diagnostics, and my colleague Neil King's work on vaccines for coronavirus. And one of the things that was exciting that you just heard about in the previous talk is how fast the world has been able to collectively move to make countermeasures. And so in our case, after the sequence was released, we first built models using Rosetta of the spike glycoprotein, and then we sort of substituted that with the cryo-EM structure and then the crystal structure as those became rapidly available. So I'm going to begin by telling you about antivirals. And this is really the same idea that you just heard about in the previous talk, to block the interaction between ACE2 and the receptor. Now, in the very nice talk you just heard, the idea was to take part of the ACE2 as sort of a mimic to block the interaction. And what I'm going to describe today is sort of a complementary approach where we instead make proteins that bind to the virus but aren't ACE2. So actually, the first approach is sort of a hybrid. So here we took one helix from ACE2, and here's the whole ACE2 molecule, which we just heard about, the optimization of this for binding the RBD. And so what we did instead is we took this helix out, and then we built a small 3-helix bundle around that helix. So we added 2 more helices and then optimized this for rapid folding. So this is similar to the approach you heard about earlier. The difference is that this is a much smaller protein because it just has the helix and a little bit of buttressing. We also generated very large numbers of 3-helix bundle scaffolds. So these are completely unrelated to anything that exists in nature. They're just built from scratch on the computer. And then we docked them against the, this is again the receptor binding domain of the virus, and then identified those that were the shape complementary, and then redesigned designed the residues at the interface to maximize binding affinity for ACE2. Now, it was interesting when we and maybe a little bit counterintuitive at first, that when we did this, the binders that we got from this approach were, perhaps not unsurprisingly, in the range of affinities of ACE2, so on the order of 20 nanomolar KDs. Whereas the ones we got from this approach, even though we were starting from scratch, we got they had much higher affinities. In fact, they were below what we could measure on our biolayer interferometry machine. So better than 100 picomolar for the best ones. That was after some sequence optimization at the interface. So we've become more, these ones have become more interesting recently with all the escape variants. And again, this was alluded to in the previous talk. So here are our sort of images of the two proteins I'll be talking a lot about. This is the one that came from the second approach. So you can see this is a 55 residue protein. This came from the first approach. So this is the ACE2 mimic. It's 75 amino acids. You can see this protein is very, very stable. And this is the one that binds really, this is one of the ones that binds really, really tightly. So this is a cryo-EM structure of the de novo binder bound to the spike glycoprotein. And here you see to give you a sense of scale, here's the whole trimer in surface view, and then the little ribbon things are 3 copies of this little binder bound. And what's really exciting from the computational design point of view is that if you compare the crystals, this cryo-EM structure to our design model. So our design model, we made this thing up basically out of thin air. We made all these little 3-helix bundles, then we docked them against the spike protein and designed the interface. And when we look at those computer models compared to the cryo-EM structure, you can see it's really close. Here's the overlay of the backbone, and even at the level of the side chains, it's really, really close. So we were very excited about that. So this is now protection of cells in culture from the virus, and this is that compound there, the one I just talked about, the LCB1 in purple, and it is it's an extremely potent inhibitor. Its IC50 is 0.15 nanograms per mil, so it's actually the most potent compound that's been identified to date that I'm aware of anyway for neutralizing the virus in cell culture on a per mass basis. The 15 picomolar is comparable to antibodies, but these small proteins at 55 amino acids are about 1/20th the size of antibodies. So our collaborators have done quite a number of animal experiments. This is one from Dan Bruch's lab, Harvard, where they basically took the protein, which is as I said, really, really stable. It doesn't melt at 95 degrees, and simply gave it to hamsters as a nose drop 24 hours in this experiment before they were infected, and then they got the virus, and those animals that didn't get the virus got very sick, whereas animals that got this protein in their nose were largely protected. The blue animals didn't get any virus at all. And this is consistent with the idea that this protein binds so tightly to the virus that any virus that's around will just get neutralized in the nose. So one of the questions about de novo designed proteins is whether they would be immunogenic. And so this experiment by Brett Case and Mike Diamond's group at WashU, they gave the protein in the nose over multiple days to the human ACE2 mouse prior to exposure to the virus, and they found that it still protected animals. These are animals that didn't get the protein but got the virus, and these are ones that did get the protein. And you can see that they are quite protected. And when they compared the amount of virus in the animals, it was far lower in the group of animals that got the protein than those that didn't. And so this looks really good. In data I'm not going to show you, they've in the human ACE2 mouse model, they found protection as far as 4 days in advance and 2 days afterwards. And the protein can also be fused to FC, as was mentioned in the previous talk, for extending the half-life. And there it functions both before and after prophylactically and therapeutically in animals. So there's obviously issues about escape variants coming up, and this was also alluded to in the previous talk. What we've done is we can take these very small proteins and now connect them in various ways. We can make triple-headed versions with three of one domain, 3 copies of one domain pointing down, that sort of land on the virus, or we can simply chain different domains together with flexible linkers. This is work from Andrew Hunt and Mike Jewett's lab at Northwestern. And what we find is this is looking at the off rate from the virus of these compounds after different lengths of time. And these are the proteins that I've been telling you about, the small single domains. You can see they come off after you wait for 14 days. But these multivalent versions are just on there. They aren't coming off at all. So the off rate becomes extremely slow. So Brett Case has been looking at these proteins, their effect on escape mutant. And this is the cell-based neutralization assay. So these are different configurations, trimeric configurations. This is the one that was the ACE2 mimic, that little 3-helix bundle. And you can see that these are sort of 3 of the major strains, the Brazil, UK, and South Africa strains. And you can see we're still getting picomolar, sub-nanomolar neutralization of virus for the 3 top constructs here. There's actually relatively little change from the neutralization of the original wild type. So we're pretty excited about this because here we have single compounds that are looking almost escape resistant. We also have other collaborators who have been looking at, in the pseudovirus model, at escape, and they are having very hard time finding escape versions for escape variants that can escape these terms. So one of the exciting things about de novo designed proteins, in addition to their small size and high stability, is they can express at really high levels in bacteria. So here's a whole cell lysate expressing that protein that was all the animal experiments were done on, LCB1, and you can see it's like the major protein in the cell. This is following expression. It is a standard T7 expression vector. If we heat the cells, then a lot of the most of the E. coli proteins just precipitate out because they're not meant to be at high temperature. But our protein, since it was again, it's very stable at TM of 95 degrees, stays in solution, and so we can purify it out very, very easily, and so we believe the cost of goods of making this kind of compound could be very, very, very much cheaper than an antibody, because you just basically, when you grow up E. coli, they just become bags of this protein. You can get them to release the protein just by heating them up, and then they pop and release the protein. So we're now excited. This protein is headed for clinical trials. So we are working on trying to figure out the optimal delivery mode, and what we're also working on. So they're sort of excited about getting this into the clinic as soon as possible, as a sort of a backup, because we think this, to treat disease, and then also as these proteins look pretty much escape resistant, in case other variants show up, these could be really useful. And we're also trying to improve the whole computational design pipeline so that we can make compounds like this in just a few weeks following the emergence of, if a new threat emerges. And we're learning a lot at really all stages in this process, even how you sort of work towards go through the preclinical studies to go to clinical trials. And so by sort of taking all this knowledge together, we think we can really streamline the computational design process, the experimental testing, and so forth. Okay, so now I want to switch to diagnostics. And here we have been designing sort of protein devices, sort of molecular devices that respond, that basically have an off state, which is shown on the left here. Here there's a cage and a key, and in the off state, the cage, this system is dark, but in the open state, there's a light is emitted. And this is kind of a similar concept to what you heard about previously. So the way this works is there's part of, I meant in the last talk, there's part of split luciferase in the small bit in the cage and the large bit in the key. So when the cage is closed, the small bit can't interact with the large bit, so it's dark. But if something binds, if there's a target present, then the thermodynamic driving force of the binding of a binding domain embedded in the latch for the target actually drives opening of the switch. And let's see. So what we, in this case, what we did is we put that the highest affinity of our designed coronavirus binding proteins that I just talked about inside the cage. And so what happens when this thing is dark in the absence of the spike, because the small bit can't combine with the large bit, but in the presence of the target, this drives the opening, and you can see the data on the right. So it's dark, and then you add the RBD or the spike protein, you get this rapid increase in signal. And we can read this out on a plate or on paper, and we can basically make these devices that recognize many different targets by simply incorporating different caging different binding proteins, design binding proteins inside these cages. And so in this experiment, these are just different sensors that we built, and each one of these, for example, this one, the third one that says troponin on in it. This is what we're doing is we're adding troponin to a set of sensors, one of which was designed to open in the response of troponin. The others were designed to open in response to all the other compounds on this list. And in each case, you can see we're only getting opening. We're only getting signal from the design sensor that was made against that target, and those are in magenta here. And one thing that's kind of cool about this format is we can tune the dynamic range over a pretty broad range. So this is here you see the response to of when you add troponin to the sensor, and you get this big increase in the amount of light emitted. And then on the right panel, you see that by changing the amount of sensor present, we can actually change the sensitivity to troponin, and so you can actually quantify the amount of response over a broad range. So, as was mentioned in the previous talk, we're very excited about using the sensor to detect responses after vaccination. And so the basic idea is our sensor is in equilibrium. So if we take the sensor I showed you just on the two preceding slides, the one that senses the spike, if we add the RBD or the spike, the sensor will open. That's in panel B here. And then if we add antibody, it competes out the RBD, so it can no longer binds the sensor, so the sensor closes, and we get a reduction in signal. It's in the bottom right. And so we can use this to quantify responses in serum. And this just shows an example using pre-immunized mice. Those are in C. Those are the controls on the first plot on the bottom. And then then we have mice that have received a vaccine candidate. I'll tell you about in just a moment. And you can see they have increasing amount of signal. And what's nice about this is we can actually correlate the amount of signal, which is on the y-axis in this plot on the right, with the amount of neutralizing titer that's been independently measured for these. So you can see there's a good correlation. And as was pointed out in the previous talk, these kind of tests are much faster than more traditional tests, because you're just monitoring light emitted. So we're also trying to figure out now how to develop this further. So the last area I'll talk about with regard to COVID is vaccines. And this is, so for a number of years, we've been designing proteins that self-assemble into icosahedral nanoparticles, as shown here. So this one's made out of two components, a pentamer and a trimer, that are designed such that they come together to form this icosahedral structure. And this works really well. We've made a large number of different nanoparticles with different geometries, and we've solved structures of quite a few of these, and they come out very close to the design models. And importantly, they're very stable and very robust and very homogeneous. So this is just sort of straight out of E. coli, you can see that we get these very homogeneous populations. And it's been possible to display a large number of different proteins on their surface. And so my colleague Neil King at the Institute of Protein Design here, the Institute for Protein here at the University of Washington, fused the RBD of the glycoprotein to the virus and made particles that look like this. So this is the RBD fused to one of the nanoparticles I just showed. And these are, again, very, very homogeneous structures. And these now look to the immune system more like viruses than just the spike trimer does. So the spike trimer is the basis for the mRNA vaccines and almost all the vaccines that are that are in the clinic now that are being used. And so what Neil's done is to compare the responses against just the spike trimer shown here to the responses against the nanoparticles shown here, and you can see they're much, much higher. This is very exciting. These actually already have this is one of the leading candidates in sort of the second generation vaccines you've heard about, and clinical trials started on these a few weeks ago. So it's a very exciting time. So I'll want to acknowledge the people who did the work I've told you about so far. So Longjin and Brian did the designed all the antiviral mini binders, working with Ina Goreshnik, and we've had really great collaborators from a number of people for the in vivo experiments. That was Brett Case, and did all the mouse experiments. Dan Baruch's group did the hamster experiments. Lexi and David solved the cryo-EM structures I showed, and Andrew Hunt in Mike's lab did all the did an amazing job piloting, identifying the best configurations, multivalent configurations of these binders. The diagnostic work was done by Alfredo and Andy and Byung-ha, and the vaccine work done by Neil King's group and the collaborators, including David Wiesler, and all this was done closely with my colleague Lance Stewart. Now, if I have a few more, how am I doing for time? I could talk a little bit about deep learning if.
Yeah, you're good on time, Dr. Baker. Feel free.
Okay, all right. So I just thought, so everything that I've told you about today is based on the idea that proteins fold to their lowest energy state, and as I said, we've developed this program called Rosetta, where we're trying to find when we make up these brand new structures like the coronavirus binding structures, and then we come up with sequences whose sequences which have very low energy in those states, and we confirm that according to the computer anyway, according to Rosetta, those new design structures are the lowest energy states for those sequences. So so that's sort of that, and we've been developing this approach for a long time. And again, it's very physically based. So the idea is that proteins fold to the lowest energy states. You calculate the energy of, you have a program which calculates the energies of different states of a protein, and so you have to then you come up with a sequence whose lowest energy state is the structure you want to make. But as you're all aware, deep learning is becoming increasingly powerful, and so we were wondering, could we actually make proteins completely from scratch, not using this energy-based model, a physically based model, but instead using deep learning? And I'm going to show you how we have been doing that. So we have been developing methods for predicting structure from sequence using deep learning, and many of you have heard about the advances that AlphaFold has made in this area. And so this is what I'm describing is very general, assuming that you have a network for predicting sequence structure from sequence. Basically, what we do is we start with completely random sequences. And then we predict what the structure is for those completely random sequences. That's in the step 0 column here. And at that stage, they're not predicted to really fold at all. This is a contact map. So residue, so it's the number of residues by the number of residues, and a dot indicates a contact between residues. So if you take a random sequence, it's unlikely to be predicted to fold to anything. And this is using the structure prediction method we're using here is called TR Rosetta. But it's just a deep neural network that takes a sequence in and predicts the structure. So what we can now do is optimize the sequence and what we're optimizing here is just the extent to which it's predicted to fold to some structure. We don't care what structure it is. The extent to which, this is now another contact map, and you can see there are many, many more predicted contacts. So we're basically trying to maximize the contrast of the degree to which specific contacts are predicted. And so if we start with different random sequences and simply optimize for folding in this way, we get very different structures out, and pictures of them are shown on the far right here. And so when we do this, and we run this many, many times, we generate all kinds of different structures. And so this is parallel to, probably many of you are familiar with the idea of network hallucinations. So if you have lots of images of cats on the internet and you train a classifier to predict, to identify cats on the internet, you can then get it to hallucinate images of ideal cats. None of them is really an actual cat, but they look like cats. And that's just the same thing with our hallucinated proteins. They look like proteins, but their sequences and structures, when you look in detail, are not those of any actual protein. And we can confirm that these things actually fold by taking their sequences and then predicting the structure using Rosetta. And remarkably, Rosetta does predict that these things actually fold. And when we make the proteins, we find that they fold in the lab. And we have structures now of several of them. So this is, and here you see comparison of the predicted structures and the and crystal structures for a 3-helix bundle here, and here for a more complex alpha beta structure. And again, it's quite neat that they come out the same. So now we have this completely new way of making proteins, which there was no reference to energy at all. It's more of like it's like this network has this sort of pattern identified as this sort of deep knowledge of protein structure. It's trained on the whole PDB, and it lets us now make brand new proteins. So there's some nice things it lets us do is with this model, we can take a sequence and predict not simply compute the energy for a particular conformation, but predict the probability that the sequence folds to that particular structure relative to all other structures. And that is that's something we couldn't do with Rosetta. And so we can actually explicitly maximize the probability the sequence folds to the structure that we want. And then the final thing I'll just note is this free hallucination problem. In this case, there's not really anything that's driving it towards a that you can't really encode function in it. But the way we're encoding function now is you specify some part of the protein that you want to have a particular geometry. So, for example, for the coronavirus case, you could specify that you want to preserve the geometry of the spike binding interface, and so you might want to mimic parts that you might want to mimic that ACE2 helix, for example. So we can basically fix those parts, and then we can basically hallucinate everything around it. So the program just comes up with all these different solutions for proteins that have that particular constrained region. And this just shows some examples of proteins that we've designed that do this. So this is a mimic of a major player in the complement cascade called C3D, and here's some little proteins that mimic it. Here's a mimic of part of insulin, and we can do this with beta sheet proteins too. So these are beta sheet proteins that mimic an interaction with CTLA4, a major cancer target. And so it's sort of a fun new thing, so I thought I'd share that. So this was developed by Ivan and Sergey, the computational method. Sam and Timuka did the experimental work for the free hallucinations. Chris and Basil looked at this ability to explicitly maximize the probability of a sequence, not that sequence folds, not just its energy. And Doug and Sydney and Zhu did the work on the hallucination. So thank you very much for your attention, and look forward to talking about taking questions later.
Very cool. Thank you, Dr. Baker. As a reminder to our audience, you may ask questions at any time using the Q&A tab. Our panel will have a chance to address these during the Q&A session. Our next speaker is Pamela Silver, an Adams Professor of Biochemistry and Systems Biology at Harvard Medical School and the Weiss Institute for Biologically Inspired Engineering. She received her BS in Chemistry and PhD in Biochemistry from the University of California. She is an established investigator of the American Heart Association, a research scholar of the March of Dimes, and an NSF Presidential Young Investigator Award recipient, Claudia Adams Bar Investigator, and NIH Merit Award recipient, the Philosophical Society Lecturer, a Fellow of the Radcliffe Institute, and was elected to the American Academy of Arts and Sciences. Silver is among the top global influencers in synthetic biology, and her work was named one of the top 10 breakthroughs by the World Economic Forum. She serves on the board of Internationally Genetics Engineering Machines, IGEM, competition and is a member of the National Science Advisory Board for Biosecurity. She led numerous projects for ARPA-E, IARPA, and DARPA, and co-founded several biotech companies, including Kula Bio, and serves on numerous public and private advisory boards. Dr. Silver?
Thank you very much. This has been a great seminar, great meeting so far. Very exciting. I'm going to talk about a couple projects that were actually in progress before COVID, but have direct application. I also want to emphasize that what we do is we tend to take things from nature and design around them to affect the functions we want. So I'm going to begin with a question that we started out asking, as I said, about 2 years ago, as part of how can we predict whether a particular emerging pathogen, how severe it might be. And so interestingly, COVID came along as a real world stress test, even though we had been into this project for already about 2 years. So the premise that I'm going to begin with is that can we use certain functions, things that happen when viruses infect cells, as a way to deduce how potentially bad that virus might be. And the thing that viruses do, obviously, they infect cells, as you've heard a lot about in the first 2 talks, and there are these physiological barriers. But what happens is that your cells will elicit an innate immune response, and what viruses do is they compromise this response. And so this then impacts your ability to respond to the virus. So what we wondered is, could we develop a relatively high throughput method to analyze how viruses affect innate immunity, which genes are involved, and then implement that to test whether or not that was a predictor of how pathogenic a particular virus was. And there were already intimidations of this in the literature, so we thought that we were on to a reasonable hypothesis. So to sum up, what I'm saying is that normally a virus would enter a cell and replicate, but the host innate immune response would prevent that from happening. But the viruses themselves encode inhibitors of the innate immune response, which then allows the virus to propagate. So as I said, we had set out on this project several years ago to ask the question, does the degree to which a virus or all viruses suppress the innate immune response predict their pathogenicity? And what we were doing was developing high throughput assays and also synthesizing genes from representatives of all viruses. So we actually had a library of about 1,000 viral genes that we were testing. Now, the assay itself interrogates the innate immune response, which is perhaps one of the best understood of all the signaling pathways in cells. And I've somewhat oversimplified it, but to illustrate it here. So the cells are responding to incoming nucleic acids. And this can be in the form of double-stranded RNA, either that's been endocytosed into an endosome, or free double-stranded RNA, or free double-stranded DNA. And these pathways are extremely well characterized with various signaling molecules in their networks, but they culminate in the translocation of key transcription factors to the nucleus, NF-kappa B, IRF3, and then there's also these produce interferons, which then work on receiving cells, and the transcription factor STAT1 is translocated into the nucleus. So I have years of studying nuclear translocation of proteins, so this was already a foundation by which we knew that we could build high throughput assays. In particular, we set out to look at the translocation of IRF3, NF-kappa B, and STAT1 into the nucleus in response to these either the nucleic acid stimuli or interferon. So this is the fundamental of our assay. And here's an example of how robust this signal is. So we developed a cell-based assay where we do high content imaging. And as I said, this is extremely robust. So these are cells where we are looking at the localization of either IRF3 or NF-kappa B. We're using poly-IC as a surrogate for RNA. And in the absence, these proteins are in the cytoplasm. But when you add poly-IC, you can see that all the cells, you get robust nuclear translocation of these transcription factors. So that is the foundation of the assay. The second foundation is this fact that there are known viral proteins already that will inhibit these pathways. And two of the classic ones are the influenza NS1 protein and the VP35 protein from Ebola. So we already had control proteins where we express them in cells. And again, looking at the IRF3 localization, we can see nuclear localization in response to stimulation, but when the cells are expressing Ebola VP35, there's a dramatic inhibition of the nuclear localization. So this difference is an easy readout for high content screening, and this is the basis on which we developed this assay. So the general procedure is, well, to take your favorite gene, co-transfect it into cells with a marker to delineate the transfected cells, stimulate, and then image. And as I said, we had synthesized a library of 1,000 viral genes to get a sense of what the performance was for this assay. And we can then quantitate the degree of inhibition using this, using our image analysis software. So we were doing this and it was going fine. And then in January, the sequence of SARS-2 appeared on the internet. I want to just comment that it's amazing how fast we all took that information and went with it, even though it was first posted on social media. So I think that's an interesting observation to think about. So we immediately decided to order genes, have genes synthesized in mammalian expression vectors that not only corresponded to SARS-2, but we decided, and I think this is key to this experiment and to our goal of seeing what the predictability was of our assays, was to synthesize the genes from SARS-1 and MERS, as well as other coronaviruses that are not as serious as SARS-1 and SARS-2. So we designed these, and this gives you a sense of our timeline. We ordered these on 2/20 from Twist, and I think we were sort of lucky to be one of the first to put in an order for these genes, and we got them pretty rapidly. Just as an aside, this served as a great resource for the Boston community because subsequent orders were slower, and so we were able to distribute these genes freely to the community. So it was a great resource. Now, our screening began just as our labs shut down. We were able to keep a skeleton crew there because this was COVID-related work. But I'll say one thing about equipment failure during a shutdown is quite catastrophic. So that probably slowed us down by several months. However, we were able to gather our first data sets on all of these genes in June and started repeating and analyzing the data. So this is what a hit looks like. So this is a cell that carries the gene of interest. It's GFP positive. The surrounding cells do not have the gene of interest. So when you stimulate them, and in this case, look at NF-kappa B localization or IRF3 localization, the surrounding cells are the controls, and you can see the GFP positive cell, there's really dramatic exclusion of NF-kappa B and IRF3 from the nucleus. So as I said, this is kind of the perfect assay for doing this kind of high content, semi-high throughput screening. So this is what the data looks like. And there's a couple things I want to say about this data. First of all, and probably most importantly to us, was that if we looked at which viruses the most genes were coming from that had an effect on the innate immune response, regardless of the gene we looked at, SARS-2 was always the winner. So just by our original hypothesis that we could predict the potential severity of an emerging pathogen, in a way, we accomplished that. Now, some of you who are very familiar with these viral genes might be looking at this data in some detail, so I just want to highlight a few things that probably more for the experts. NSP1 is a positive hit, and NSP1 is strongest from SARS-2. NSP5, which is the protease, scores as a positive, and that's kind of an interesting result because there's, of course, a lot of interest in making inhibitors to the protease as a therapeutic. So this actually gives us an in vivo test for the action of NSP5. If we inhibit it, does it still elicit an innate immune response or not? I want to mention NSP3 as it's a huge gene, and many have been unable to assemble it, and we finally did. So this was quite an accomplishment. So we were able to analyze the intact NSP3 as opposed to the parts of it, which is what you often see in many papers. I think those are the main highlights I want to make about this data, but I want to re-emphasize the take-home message that if you had done this a year ago, you probably would have been worried about SARS-2. All right, so we could call that assay a mechanistic-based assay, so we sought a more agnostic assay that would not be so connected to specific mechanism. And this builds on work that we have been doing for a couple years now with John Glass at the Venter Institute, where we're very interested in how to introduce large pieces of DNA into mammalian cells with a long-term interest of, for example, building human artificial chromosomes. And so what we've done, we did this several years ago, was to develop a vector that it's a yeast centromere plasmid. So it lives in yeast, and it encodes, in this case, an M-cherry protein and a GFP protein under the control of a mammalian promoter, the CMV promoter. And what we found is that if you convert these cells to spheroplasts, so you get rid of the cell wall, they will quite efficiently fuse with mammalian cells. And you know the fusion has occurred because of the presence of this soluble M-cherry in the cytoplasm of the mammalian cell. And then the DNA will be taken up by the mammalian cell nucleus, and then in this case, the GFP will be expressed. Now, we also showed several years ago that the efficiency of uptake is enhanced if you have mitotic inhibitors, and you can transiently treat cells so the nuclear envelope is broken down, and you will see more efficient uptake. Now, to get to this idea of developing a second assay where we look at the effect of these genes on viral production, what we did was to engineer an HSV1 genome into a yeast plasmid, a yeast vector. So this is now the entire HSV1 genome, 152 kb propagated in yeast. And in this case, the idea is that this is in a yeast cell, and if it's delivered to a mammalian cell and it can propagate, it will produce virus, and then you can assay that virus by plaque formation. And so these are with HEC293 cells. There's no virus produced. This is a negative control. But if you have yeast that have the HSV1 genome, you see plaques. And this is the interesting, important part about this experiment. If we do this with HeLa cells, which have a more intact innate immune system than HEC293 cells, sorry about the typo here, you see no HSV1. So remember that. So then we took this experiment one step further, and we realized that we could then fuse mammalian cells with two different yeast cells, one carrying the HSV1 genome, and the other containing libraries, or for example, containing VP35 or our favorite viral gene expressed in a yeast cell, and that these could be fused together to the same mammalian cells. And if the protein that's delivered then suppresses the innate immune response, it will then allow propagation of the HSV1 genome. So this experiment then is done in cells that are not permissive for propagation of HSV1, such as HeLa cells. And what was amazing about this is that it actually worked. So here is the data I showed you earlier. This is the data, this is the control with 293 cells. So they produce HSV1. There's no impact of adding VP35. With HeLa cells, they don't produce HSV1. But if you co-fuse them with yeast cells making VP35 from Ebola, which we already know suppresses the innate immune response, we see viral production. So again, this is our control for what we are setting out to screen for. So we built a library, again, of all those different coronavirus test viral proteins, and then took it through this screen. We streamlined the screen so that instead of looking at live virus, we looked at expression from a late promoter, so we have a fluorescence readout. So this can be done in a plate reader. And so this, again, is the cumulative data. These boxes are indicators of a positive signal from the late gene showing that HSV1 is indeed propagating. And again, as in the prior experiment, SARS-2 is the winner. And the other genes from the other virus generally often score negative in this assay, or there are fewer of them. So again, this is an agnostic assay to mechanism, except that it is allowing virus to propagate. So we like to think that this can be a predictor for how potentially bad an emerging virus can be. And we sort of had a real world stress test with COVID. And so it's interesting to think, if we had known a year ahead, could we have predicted this? And the other thing is that this elucidates potential new targets for therapy, as well as new ways to screen for therapeutics. All right. How am I doing on time?
You're doing good. You're doing good.
Okay, I have one last, then more generalizable synthetic system to talk about, which, as I said, is more generalizable and draws on nature. And it has to do with this idea of, could we build a living biosensor for pathogens in the environment? And of course, we can think of lots of reasons to do this. And we would like to make something that, again, would draw on the strengths of what nature has on offer and might be cheaper and easier to produce. So we would like to build a system that's cheap and scalable, would have no supply chain problems, and would be highly specific, but something that could be added, say, to water and give a rapid readout. So we want to use the ability of cells to sense the presence of specific DNAs. So again, we turn to biology and we turn to Bacillus subtilis, which is naturally takes up DNA from the environment, and is, again, a very well understood organism in terms of engineering. So our system looks like this. We want cells that can take up DNA, recognize it, read it, produce a signal that it has seen a particular DNA, and amplify that signal so that we can read it out. So this is the general schema for what we're trying to make. And so let me show you these different modules. So first of all, with regard to uptake, here there's a GFP sensor that's recording how well DNA has been taken up by subtilis, and we were able to create a B subtilis strain that where over 70% of the cells now take up DNA from the environment. So that is well within the range of the efficiency we would need. Now, to amplify the signal, we're going to draw, of course, on DNA recognition by dCas9, but we're going to use the following system to amplify the signal. So in B subtilis, there are receptors that have kinase domains. So these are just like serine threonine kinases that occur in higher cells. And in order to be active, this kinase has to dimerize. And in a B subtilis cell, when there's when this receptor binds to its ligand, the kinase domains dimerize, they phosphorylate a transcription factor, which then activates target genes. So we wanted to co-opt this kinase to be our amplifier of the signal. And so what we did was to fuse the kinase domain to a dead dCas9, which has no activity, but except that it will bind the guide RNA and recognize DNA. And the general idea here is that when there are two guide RNAs that recognize two adjacent sequences on the DNA, this will promote the dimerization of the kinase. It will activate its transcription factor, and hence there will be a transcriptional readout. Now, there's a lot of parameters that have to go into this design. You can imagine the length of the linker being important, the distance on the DNA, etc. We optimized all of those, and we also we did two things. One is we looked for the activation when the kinase is scaffolded on the DNA. And we also looked for its ability to repress an mCherry gene as an internal control for whether or not it's really recognizing its cognate DNA. So here is the data on repression. So we can find the perfect conditions where you have two sites spaced by 62 base pairs at 20 amino acid linker, and you see repression of the target mCherry gene, and simultaneously you see, and I should say this is over growth, you see activation over background only when you have the correctly paired guide RNAs. And this can be actually taken down to the single cell level as a readout. So these are cells where the kinase is active, and you can actually see at a single cell level which cells are producing the correct kinase signal. So this is the system that we built, taken together. There's been a lot of improvements along the way, but I want to just end by saying that we actually, again, did a form of a stress test where we took blinded samples with different levels of, in this case, the TED-R gene mixed in, and we did a plate assay using these using our sensor strains, and actually basically hit a home run in that we detected the TED-R gene, and we could deduce the level to which it was present in the blinded samples. All right, so to end with, where we can go with this, a cell can sense more than, you can put different sequences in one cell. This is relatively fast. It's pretty robust to contaminants in the samples. And as I said, there's really no supply chain problem. You can grow up bacteria. They're really cheap. So that is what I wanted to say. And I will end by acknowledging my wonderful colleagues, in particular, Erica Olson, who was the postdoc that really had the insight to go ahead and order all those coronavirus genes. I wasn't really thinking about it. And the team she led to make that all happen, together with our colleagues, Debbie Marks and Nathan Rollins, who we've been working with on the Big Virus Project. And then, of course, John Glass at JCVI, who has been our long-term collaborator on this project. And the living DNA sensor was the brainchild of Elizabeth Libby, who was a departmental postdoc and now has her own lab at Northeastern. So thank you very much.
Awesome. Thank you, Dr. Silver. Remember to submit your questions for our speakers in the Q&A tab on the right of your screen. We will have a chance to address them after our next speaker. And that speaker is Jim Collins, the Centromere Professor of Medical Engineering and Science and Professor of Biological Engineering at the Massachusetts Institute of Technology, MIT, and a member of the Harvard-MIT Health Sciences and Technology faculty. He is also a core founding faculty member of the Weiss Institute for Biologically Inspired Engineering at Harvard University and a member of the Broad Institute of MIT and Harvard. Collins is one of the founders of the synthetic biology field. His research group uses synthetic biology to create next-generation diagnostics and therapeutics. Collins's patented technologies have been licensed by more than 25 biotech, pharma, and medical device companies, and he helped launch a number of companies, including Synologic and Sherlock Biosciences. He has received numerous awards and honors, including a Rhodes Scholarship and a MacArthur Genius Award. Collins is an elected member of all three national academies, the National Academy of Sciences, the National Academy of Engineering, and the National Academy of Medicine. Dr. Collins? Dr. Collins, you might just have to click that microphone icon at the top. All right, let's see. So if you click that microphone icon, it should turn on your speaker. I think, Dr. Collins, can you hear us? Okay. So just have to reach up to that icon and just click it on to give access.
It may just take a moment to load. Sorry for the delay, everybody. Hi, Dr. Collins. If you can hear us, you should just have to click that microphone icon at the top of your screen to share your computer audio. Dr. Collins, if you can hear us, we just need you to click that microphone icon. You can also try clicking on the camera icon if that's not working for you. Should also turn your audio on for us.
You may also call in, Dr. Collins, if the computer is giving you a little bit of trouble.
Sorry for the delay, everyone. We're just working through some technical issues here, and we'll be back momentarily.
Hello.
All right, we got you. You sound loud and clear to us, Dr. Collins. Can you hear us?
I can hear you very well.
Perfect. Thank you. Please feel free to begin.
Okay. Well, I want to thank the organizers for having the chance to present our work to you. What I want to do is share what we've been doing in synthetic biology and deep learning to fight pathogens, including efforts to go against COVID-19. As shared by Pam in her great talk just before mine, synthetic biology is in a maturing field. It's been around for about 2 decades. And the prime focus is on using engineering principles to model, design, and build synthetic gene circuits and other molecular components, including proteins of the type David Baker spoke about, and using these to rewire living cells, endowing them with novel functions that can provide them with the ability to address a range of applications. We and several other groups helped launch the field in the late 90s when we recognized that just as an electrical engineer will come up with a schematic for an electronic or electrical circuit, we as biological engineers could come up with a schematic for a wet or biological circuit. And whereas the electrical engineer will seek out electronic components, resistors, capacitors, inductors, and solder them onto a breadboard, we recognized that we could seek out molecular parts, genes, promoters, other bits of DNA, RNA, and use the enzymatic tools of genetic engineering in a cut and paste fashion to create plasmids of DNA and other constructs that can be transferred to cells and then tested to see if they function as desired. The first system that our group designed and built in this manner was a genetic toggle switch, and this was work done with Tim Gardner, and it was motivated by toggle switches, also called flip-flops, some electronics, that are very simple memory elements that can be flipped between 0, 1, or on-off. The circuit schematic we came up with is captured in the bottom of this slide, where it was essentially 2 genes, each being constitutively expressed, but arranged in a mutually inhibitory network. And the system can be set up so that a bistable system want to exist in one of 2 states. Either state A, gene 1 is on and gene 2 is off, or state B, gene 2 is on, gene 1 is off. And you can flip them between those states by translating, delivering chemical signal or environmental signal, creating now programmable memory. We actually initially did mathematical modeling and then created a range of plasmids that could function inside E. coli, and in a matter of months designed a number of functioning bistable toggle switches, captured in the right, where you could start them in an off state, flip them to an on state. They would stay in the on state indefinitely until you'd flip them back to the off state, which they will also stay indefinitely until you're ready to flip them back to an on state. The system had intrinsic nonlinearity that beautifully provided a sharp switching threshold, as captured down below, which opened up possibilities for sensing and diagnostic applications along the lines of what Pam just shared. This work got a lot of excitement around the idea maybe we could begin thinking about programming bacteria to replace computers. But that really was not the right direction for the field. And instead, the field, starting around 2004, really began to move towards the idea of creating programmable cells, so living technology, that along the lines of what Pam just shared, where you could take now synthetic circuits, introduce them into living cells, have them coupled to different input and output pathways, where now you could program cells to sense their environment, make a decision, and then act on their environment. And these living technologies, living diagnostics, living therapeutics, are impacting broad swaths of the economy, including and in particular medicine. At the very end, I'll return to some efforts in this spirit for COVID-19, but want to shift gears slightly to work we did going back now 7 years ago. And this was efforts around moving synthetic biology out of the lab and into the field by taking advantage of cell-free synthetic biology. This was work being done by Keith Pardee in my lab. And Keith was then a postdoc. And as many of you know, with cell-free systems, you can basically take the machinery of a living cell out of the cell and play with it in a petri dish or test tube. This machinery would consist of DNA, RNA, molecular machines like ribosomes, and biomolecules like ATP, nucleotides, et cetera. These have been used for decades in molecular biology. Keith had the idea to see what would happen if you took a cell-free extract along with a synthetic gene circuit, it might be a sensing element of the type Pam just presented, and spotted these on paper and freeze-dried it. What would happen? Well, what Keith found was that subsequent to the spotting and freeze-drying, sometime later, if he rehydrated that freeze-dried spot, he could reactivate the cell-free extract. He could reactivate the synthetic gene circuit so that you could have transcription and translation, and that freeze-dried spot would then function as if it was inside a living cell. We got excited then about how this could be applied in many different directions to address pandemics of various sorts, and particularly got excited how you could potentially use these freeze-dried systems to create inexpensive, field-ready, paper-based diagnostics. Captured here in the bottom left are sensors that we developed with Peng Yin and Alex Green at the Wyss Institute called Toehold Switches. These are basically repressed mRNA elements where you can engineer the system so the ribosome binding site is at the end of a stem structure, preventing the ribosome from docking to the RBS. That stem structure can be created with a sequence that corresponds to, in a complementary fashion, to a pathogen sequence you might want to detect. If that pathogen sequence is present in the form of RNA or DNA, it can then linearly interact with the stem structure, opening it up, exposing the ribosome binding site to the ribosome, enabling then the encoded gene to be translated. We initially did this to create living microarrays inside bacterial cells, but recognized with Keith's advance around freeze-dried cell-free synthetic biology that we could also use this as the way to create inexpensive paper-based diagnostics captured in the upper right. The beauty of this system is you could then encode, say, for example, LAG-Z is shown in the upper left, that when expressed, turned on in the presence of an RNA of interest, would change the color of the paper from yellow to purple. This then gave us these inexpensive diagnostics, cost less than 2 cents per sensor, did not need to be refrigerated. So you could stick these in your pocket, fly around the world, take them out and use them anywhere in the world, and open up range of possibilities. We initially looked at using these for antibiotic resistance. And captured in the bottom right, our notion was, could you use these to identify in that golden window of an hour or two the patient might have when they present in an emergency room, before being prescribed with an antibiotic, do they have a bacterial infection and is it resistant? We were able to show that you could, and we were moving towards publication in the summer of 2014 when Keith and Alex came to my office and asked that we hold up on the publication. I asked why, and they wanted to see if they could use the platform to go after the Ebola crisis. So it was not a pandemic, but it was a crisis and an outbreak in a number of countries in Western Africa at the time. And I said, of course. And what the team did was really remarkable. In a matter of 12 hours, Keith and Alex designed, built, tested, and validated 24 different sensors for Ebola. 12 for the Sudan strain, 12 for the Zaire strain. They will obviously do a number of different control reactions as well, but then embody these into a multi-well, multi-spot paper-based system that enabled them to differentiate between the Sudan and Zaire strains with outputs being provided within 20 to 30 minutes subsequent to needed amplification. Again, with each sensor only costing about 2 cents. Fast forward, we got very excited then and began to look at different applications, but in January 2016, received an email from MIT leadership asking all faculty who was working on Zika and could we coordinate efforts at MIT. We were not. We were working on extending the platform then to seasonal flu. But I reached out to Alex and Keith, who had then started their own faculty labs. And in seconds, they said, let's do this. Let's see if we can apply this to what became the Zika outbreak. And turned to a number of folks in my lab, and to a one, they immediately say, yeah, let's do this. And in a matter of a few weeks, our team was able to develop a field-ready, paper-based Zika diagnostic that involved freeze-dried isothermal amplification and NASBA step that could then enable us to detect down at very low femtomolar, which was needed for clinical samples. We were able to differentiate Zika from other fever-based illnesses in the endemic regions, including dengue. And we actually did the first CRISPR example of a diagnostic. Guillaume Lambert, postdoc in my lab, recognized we could use CRISPR-Cas9 to identify SNP differences between strains of Zika. So subsequent to the first step shown on the left on this slide, one could first identify, do you have Zika? And then if you had Zika, you could then do the CRISPR test that could then identify what strain of Zika that you have. This test was actually deployed as part of the outbreak as a research and surveillance tool in 7 different countries. And Keith Pardee has just completed an in-field patient trial in Colombia, Ecuador, and Brazil, looking at Zika, dengue, chikungunya, and yellow fever, and obtained 98 to 99% both sensitivity and specificity, which we're very, very excited about. Kind of queuing us up toward a very small example of how we use this for COVID. It's intriguing to think about this design, build, test cycle that really is at the heart of many of the efforts in synthetic biology. The challenge with this is that most of the stuff we build in biology does not work in the way we'd like when we first build it. And this leads to then the need for iterative cycling around design, which can be really time consuming. We decided to see, in collaboration with George Church, in collaboration with Tim Liu, in collaboration with Diego Camacho, colleagues at the Beast Institute at MIT, could we harness the power of deep learning to create a platform that could advance our ability to do bottom-up design in synthetic biology? And the idea was quite simple. Could we take various constructs, synthesize them into diverse libraries, sequence those constructs, and functionally characterize them? Feed these as training data to deep neural nets that could then begin to identify sequence to function relationships and composition of function relationships that we could then use the outputs of these models to infer design rules, as well as to design novel components and novel gene circuits. What we initially did in work with George's team was to synthesize 250,000 different toeholds. Those were the sensors I talked about earlier that underlied our work in antibiotic resistance, Zika, and Ebola. We did a quality check, and only about 92,000 of these met our criteria, but we then used that downsample set to train a number of deep neural nets. We compared the output of these deep neural nets to a number of popular thermodynamic and biophysical-based models. And it's not as clear in this slide as I'd like, but briefly what we found were that the deep neural nets far outperformed the biophysics and thermodynamic-based models at predicting the function of the RNA switches. And we could use the deep neural nets both to predict on-off and on-off ratios, both on functions, off functions, and on-off ratios, as well as properly classify good and bad switches. One of the challenges with machine learning writ large in AI is that its applications in biology, biotech, and medicine are often limited because the derived models are often treated in a black box fashion where it's difficult to derive and gain insight. Our team took advantage of that challenge to say, let's train the model on 2D projections of these RNA sequences for our training data, and then project the output of the model onto those 2D to gain insight into the sequences that matter for the toehold switches. And we were able to gain mechanistic insight into which regions of the toeholds mattered for their design. In a companion piece that came out in the fall in Nature Communications and efforts that were done by Diego Camacho and Tim Liu's lab, Jackie Valeri and Katie Collins came in and did 2 things. One is they actually integrated convolution neural nets, which dominated our efforts, with natural language processing neural nets in an integrated fashion that actually led to superior predictions of on-off behavior, as well as superior classification ways of getting after good or bad. But more interestingly, they showed how you could take the learned model and actually use it to both design de novo RNA switches for various applications and optimize the obtained ones. And what Jackie, Katie, and their team were able to do was actually design toehold sensors for COVID that they then could optimize and improve, and shown on the right. And efforts around these also were built upon by Keith Pardee and Alex Green that actually developed paper-based efforts to create deployable systems for COVID diagnostics. Shifting gears slightly, when we first reported on our Zika diagnostic in the Cas9 component, again, the first kind of CRISPR example of a diagnostic, Feng Zhang of CRISPR fame and my colleague at MIT in the Broad reached out and he shared that his team was working on Cas13 and wanted to see if we would collaborate with them on a different diagnostic application. And we immediately jumped at the opportunity. Cas13 is different from Cas9 in two ways. One is Cas9 goes after DNA, whereas Cas13 goes after RNA. And second, whereas Cas9 in the active form, when activated, brought to its target by its guide, will degrade with high specificity its target. Cas13, when brought to its target by its guide, will not only degrade that target when it's activated, it will then degrade any RNA in its vicinity via collateral cleavage, collateral activity. We decided to use this latter feature as a key part of this new diagnostic platform. And what we did was say, okay, let's now design guides that can bring Cas13 to its particular sequence of interest. It might be a pathogen, it might be a host RNA or DNA. When it finds it, then let's have it activated to break down RNA in the vicinity. And what we did was now for readout for the diagnostic, have quench fluorophores where the fluorophores were held close to the quencher by RNA linkers that would then be the target of the activated Cas13. With this system, we found we could get to low attomolar, so single molecule per microliter sensitivity. Going back 3-4 years ago when we first presented this work, we focused on Zika and dengue. We did work on HIV. We showed you could apply this to bacterial pathogens. You could speciate the pathogens. You could identify mutations underlying drug resistance. We were able to embody these into low-cost, easy to deploy, paper-based lateral flow assays, microfluidic systems that's captured here for looking at Zika in the field. And it wasn't limited to pathogens. We showed this system could be used to basically genotype patients. So within an hour, we could take a sample of a patient and identify the presence of different SNPs, opening up interesting possibilities for screening, for example, for clinical trials. On the basis of this early work, Feng Zhang and myself launched a company called Sherlock Biosciences. And the idea was to create a next generation molecular diagnostics company that could leverage this technology to get after new viral diagnostics, bacterial diagnostics, patient genotyping capabilities. The company was doing very well and continues to do very well, but in the midst of the pandemic in mid-February, decided to pivot to go after COVID. And what their team did, and input from our respective teams, was really remarkable. They were able to develop, within a matter of small weeks, a CLIA lab-based COVID test that had 100% sensitivity and 100% specificity against the FDA samples, on par with RT-PCR, but it was at a quarter of the cost and could give you an output in 1/6 the time. This led to an early May FDA EUA, so emergency use authorization, making this the first FDA-approved CRISPR product to hit the field, hit the market. And Sherlock subsequently teamed up with IDT for a manufacturing partnership that is focused on and capable of producing 1 million of these per week. Nepal has now adopted this test as their major test for the country, and Thailand is in discussions with Sherlock as well. Sherlock is advancing efforts now in a collaboration with Binx to create a point of care version, and Binx, along with Sherlock's interest and input, will be applying for an EUA in the coming small weeks. Sherlock, with input from our lab, using a slightly different synthetic biology construct, is now just a matter of hopefully a few weeks away from finalizing an at-home diagnostic that should be potentially quite useful as a surveillance tool. What I didn't share was when Keith Pardee made his discovery that he could freeze-dry cell-free extracts and synthetic biology constructs onto paper, was that Keith also uncovered that it wasn't limited to paper. And that is, he could actually freeze-dry these cell-free extracts and these synthetic biology constructs onto any porous medium. Could be plastic, could be quartz, could be cloth. And prior to the pandemic, effort led by Peter Nyagun, Nina Dangya, and Louis Songson in our lab, we were pushing to create a wearable synthetic biology platform that could be used by first responders, healthcare workers, military personnel, hikers, as wearables that could report on if they were exposed to a pathogen or a nerve agent, or if their body's responding in a stressful manner in producing different molecules. Our team was able to show by testing over 100 different fabrics that all of them could serve to wick up a cell-free extract along with synthetic biology constructs, including the toeholds, that could then be freeze-dried and used later when reactivated by a patient sample or environmental sample as a diagnostic. We embedded a number of different sensors, including Rabo switches that could be used to detect small molecules in the environment or given off by the person's body. Toehold switches, which I've already talked about, which we showed could be used to detect exposure to different viruses, as well as other synthetic gene circuits, which is the one shown on the bottom right, which was a universal generic circuit we designed to detect nerve agents. We were able to couple these systems with flexible electronics, optoelectric fibers that could detect the output of the synthetic circuits that could then be embedded in various embodiments that could send signals to your cell phone or to a PC, for example, at a nurse's station. We initially were focused on using this technology to create, for example, lab coat of the future that could be worn by doctors, physicians, assistant nurses around the wards to see, as they report around on patients, have they been exposed to a pathogen, and if so, track it back to the source. We also don't want to forget about the patients, and we're working on embodying these into Johnnies so that it could report on in patients. Do the patients, for example, have a pressure sore? Is the pressure sore infected? Is it infected with a resistant pathogen? As with Sherlock Biosciences, when we were faced with the pandemic, we quickly pivoted and thought about how we could harness this platform to address the pandemic. And the notion we had was to see if we could use this wearable synthetic biology platform to create a face mask-based diagnostic. And the idea was quite simple. Could you create an insert that could be added to any face mask that then could be worn by an individual for 1 to 2 hours, and the normal act of breathing, talking, coughing, sneezing will give off water vapor, will give off water particles. And if the person is infected, that water vapor, those particles, water droplets, would contain the virus that could be detected. The design we came up with was a really quite elegant one that was based on foldable, paper-based microfluidic lateral flow assay that had staged regions. We had a collection zone, a lysis zone, chemical lysis zone, a room temperature amplification zone, a detection zone based on CRISPR or Sherlock, and then an output zone. Each of these was separated by different sugar bridges to introduce an intrinsic delay. We were able to build these, test them out at the V's quite successfully, and engineered sensors, designed the sensors using various bioinformatic tools, targeting different aspects of the spike gene, spike protein, and found that the system had incredibly high sensitivity. We could detect 500 viral particles, which are relatively easy to collect after, frankly, just a matter of minutes in the face mask, and we could do it with very high specificity. So we could detect SARS-CoV-2 with high specificity relative to other circulating coronaviruses, and we're able to multiplex this so that in addition to detecting the coronavirus, we could also detect readily other seasonal flus. Shifting gears in the last set of SynBio examples, I want to give just one more deep learning example, was to speak briefly about the Moderna vaccine and to point out that their underlying technology, the synthetic mRNA technology, actually has synthetic biology roots. We were lucky enough to be part of the team that co-developed this technology with Derek Rossi, who became a founder of Moderna, in collaboration with George Daly at Harvard Med and the Beast Institute, going back 11 years ago. Our idea was not to go after vaccines, though we spoke about this technology as being potentially useful for vaccines and therapeutics in the discussion, but it was to get after stem cell reprogramming and redifferentiation. So this is just a few years after Yamanaka's beautiful landmark work. And our notion was, could we engineer, create mRNAs, synthetic mRNAs, by replacing different nucleotides so that they would not trigger a viral response in the cells, 2, that they would not be readily degraded, and 3, that they could express at a high level. And we were able to do all of those and then deliver them with lipid nanoparticles to various cells to reprogram the cells and then deliver another set using Yamanaka factors, another set to redifferentiate them. This work, if you actually look at the paper, it was a stem cell paper of 2010. In the disclosure, Derek actually noted that he was founding a company that he called Mode RNA to commercialize it. And this underlying synthetic mRNA technology, again, of synthetic biology origins, underlies Moderna's effort. And Moderna is using synthetic mRNAs to express the SARS-CoV-2 spike protein that will then be taken to basically the outer layer of the cells and expressed in the surface, triggering a very strong immune response. And as we've all seen, they've gotten tremendously powerful and very positive results. We've been taking also a slow route and a different route on the vaccines in our lab by looking at how you create, going back to kind of this idea of living therapeutics and some of the work that Pam shared, could we have live vaccines in a different form? And what our team did, in part in collaboration with some folks at Cornell Med, was explore the Bcg vaccine as basically a vehicle for COVID-19 vaccine. Here, the idea is Bcg vaccine, it's one of the oldest vaccines, been around for about 100 years, used for protecting against TB. It's based on the Mycobacterium bovis bug, which causes TB in cattle, but not in humans. Has an incredibly good safety profile, easy to manufacture, only requires a single dose, and very cheap, does not require refrigeration. Our idea was, could we engineer Bcg to express the spike protein so that you could then deliver this intramuscularly, maybe even give it intranasally. And our early results indicated that we could get very good expression. And early efforts in a mouse indicated we got a strong background immune response, but not as strong an antibody response as we'd like. And we're now exploring to see how we could boost that, and could we use this as a broader platform. Just to close, I want to briefly share that also, just as the outbreak, the COVID pandemic hit, we reported on a new effort in our lab on using deep learning to discover new antibiotics. And where it's relevant to COVID is that it's underappreciated how important bacterial co-infections are to the current pandemic. 1 out of 7 COVID patients who are hospitalized have a bacterial co-infection. And 50% of the patients who die with COVID or of COVID have a bacterial co-infection. So there's a desperate need to address both those challenges, as well as the fact that we have increasing incidence of resistance due to overuse. With Regina Barzilay, we teamed up and basically came in and created a platform where we could train deep neural nets against different data that could identify molecules that might be new antibiotics. And we identified a whole range of these, which we tested successfully, opening up the possibility that AI could really get us out of the antibiotic resistance crisis and address other challenges. So what we're now doing as part of what we're calling the Antibiotics Ai Project at MIT is not only developing deep learning models as discovery tools, but also as design tools. We show that you can train deep neural nets to learn molecular features that are associated with the desired property. We right now are going after antibacterial, but I'll come back to that in a second. Where now you could then use that to design de novo molecules with properties, and we're advancing efforts now to go after resistant Acinetobacter baumannii, a nasty bug at the top of the WHO's list in desperate need of new antibacterials. This platform is not limited to antibiotics, so efforts initially led by Regina Barzilay at MIT, my colleague, under the AI Cures effort, was to see if you could use this to get after novel molecules that could go after COVID-19. And we and others look to see if you could look at repurposing libraries, which didn't come up with as high yield as we like, but we're now looking to see how we can use these learned models kind of in a similar spirit to how David Baker addressed the problem in his beautiful talk earlier, to see if we can have these features to come out with novel molecules. And with that, I want to end and just thank this team of outstanding young folks whose work I got to share with you today. Thank you.
Thank you, Dr. Collins. The audience has submitted several questions, so let's begin the question and answer session. Jeff Glasgow will also be joining our panel. Glasgow received his Bachelor of Science in Biochemistry and Molecular Biology from Oklahoma State University, where he researched protein crystallography. Glasgow earned his PhD in chemical biology from the University of California, Berkeley, under the co-mentorship of Matt Francis and Danielle Tillman-Erck at Berkeley. Glasgow combined the tools of bioconjunction chemistry and protein engineering to create nanomaterials for a variety of applications. Glasgow then transitioned to a National Research Council postdoctoral position with the National Institute of Standards and Technology at Stanford University. Under the advisement of Mark Sullett and Jennifer Cochran, Glasgow worked on enzymatic methods for protein bioconjunction and measurement technology for characterizing bacterial translation from non-canonical start codons. Currently, Glasgow is a postdoctoral scholar in the laboratory of Jim Wells at the University of California, San Francisco, where he develops T-cell-focused immunotherapies from engineered antibodies and other proteins. During the current pandemic, Glasgow has developed protein-based SARS-CoV-2 therapeutics. Welcome, Dr. Glasgow. Our first question is for Dr. Anum Glasgow and Dr. Jeff Glasgow. Is there any particular reason why you used rosettes in this study, and why not other strains?
Yeah, we didn't actually use Rosetta cells. If it was on my slide, it was because we used the program for protein design.
Fantastic. Okay. Our next question, I'll direct first to you, Dr. Glasgow. If the receptor trap strategy progresses to the drug development stage, what would be the potential effects of ACE2 receptors in other body tissues, blood, vessels, heart? And consequently, what might be the potential adverse effects of these drugs?
Yeah, this is a good question. ACE2 is expressed in a lot of these body tissues. I'm going to defer to Jeff because he has an encyclopedic knowledge of this of this area.
Thanks, Anam. Yeah, so this is an interesting question, important for our preclinical development that we're doing. Two potential avenues of side effects could be ACE2 interacting with the wrong thing, or ACE2's enzymatic activity. ACE2 has very few annotated interaction partners, so we think like off-target binding is probably not going to be that much of a problem, but the enzymatic activity is actually a really interesting potential benefit and also potential side effect, as the current clinical trials involving ACE2 are actually using the enzymatic activity as a benefit to treat these pulmonary side effects that you get from COVID infection.
I see. Dr. Baker, on the topic of receptor traps, would you like to comment about the potential effects of receptor traps in general on other body tissues, blood, vessels, or the heart?
Oh, let's see. I thought that was an excellent answer. I don't really have anything else to add.
Great. Our next question is actually for you, Dr. Baker. Why did you decide to go to the approach of using mini proteins as opposed to targeting the ACE2 location?
Let's see. Well, we were trying to design proteins that bind to the virus, and so we wanted to make a therapeutic that would prevent infection, and so that's why we were designing binders for the spike protein, for the RBD specifically. We could have designed instead binders to ACE2, but then the previous question becomes relevant. If you have proteins that bind to ACE2, it carries out important physiological functions. And so we were concerned about that, and that's why we targeted the virus rather than ACE2. I'm not sure that answers the question.
No, that's perfect. Thank you. Dr. Silver, are you looking at using these new assays with new viruses or potentially even the new variants of SARS-CoV-2 to see if they're more pathogenic?
Yes. So, well, first of all, the new variants, mostly people are concerned about the mutations in the spike gene, as you heard earlier from the earlier talks. And so we are looking downstream of what would happen after the virus enters cells. But it's interesting because we're also taking a look at the variants, and they do have mutations in these other accessory genes, which people aren't paying as much attention to. So we are considering that.
Great. You mentioned a couple times, Dr. Silver, that if we had just looked at this 2 years earlier, we would have kind of seen SARS-CoV-2 coming. How do we get ahead of the pandemic, in your opinion?
Well, I think this is what, and I'm very excited that there's a lot of interest in this, is how do we predict pandemic potential? And I have to hand it to a lot of government agencies have actually been working in this space. so there, even though you don't hear a lot about it, this is a huge area, and I think it's going to become even bigger. And I'm really excited about that. And we have so many new tools to do that with, which I think is a lot of what you've heard about. So some of the predictions, I think, will come through things Jim alluded to, through ramping up machine learning and looking at histories of viruses, and looking at escape mutations and things like that. But having really broad-based platforms, I think, is going to be key also to assessing what the fate of a particular emerging pathogen is.
Indeed.
I think it's a huge area, and I'm excited. One thing positive about COVID is that I think it's been a giant national biology lesson. And so I'm excited that this is going to bring forward a lot of people into this space.
Indeed. Dr. Collins, can you tell us a bit more about the challenges of deep learning? It seems so fascinating, but it feels like it can go several directions. What are the biggest challenges?
Yeah, I think it comes off as being magical to many, and it's not at all magical. It's very data hungry, and you need to have diverse and appropriately good quality data. And I think you have to ask the right questions. I think you have to have the right kind of multilingual teams in place in order to really get the most out of it. And for those interested in exploring, I would say seek out the right collaborators, and most critically, view it that in biology and biotech and medicine, that I think it's best used right now as an augmented tool for our efforts. And that is that I think the biggest discoveries will really come from the efforts where we're integrating machine intelligence with human intelligence. That I don't think we're at a stage where things could be automated and we just step out of the picture, in part because I don't think we know enough about the complexity of the biology and chemistry to truly automate it yet. But I think integrating those two is where I think we're going to see some pretty interesting discoveries and developments unfolding in the next decade.
Thank you. Dr. Baker, how do you decide which proteins are worth pursuing with so many possibilities? You mentioned that you're not limited to protein folding, and you can create basically any protein structure. So how do you know which ones to pursue?
That's a very good question, and that's one of the challenges with the space, there being so many possibilities. We're really inspired by nature. I mean, there's so many amazing things that proteins do, ranging from protein-protein interactions, like you've heard about a lot today, to light harvesting, to machines. And so we're trying to design proteins really trying to design new machines. We're trying to design, new light harvesting systems, new sensors. I mean, it's a good question. I think the way that we ultimately decide is really what the people who come to my group are excited about doing, because there is a really huge universe of possibilities when you think about new protein functions. So one way to think about it is that the proteins that exist in nature evolved to solve the problems that were relevant during biological evolution. But now, of course, we live in a totally different world, and so there are a whole bunch of there are huge numbers of new challenges. And so now basically the challenge for protein design is to create solutions for those problems, like would evolve naturally if we had 1 billion years to wait. But with protein design, we can try to do it right now.
Thank you. Dr. Anam Glasgow and Dr. Jeff Glasgow, I'll direct it to you guys, beginning with Anam. You mentioned using yeast recombinant source of AC2. Do you have any plans to look at how is the glycosylation evaluated against the natural binding of human AC2 receptor?
Yeah, this is a fair point. We expressed all of our proteins for testing in vitro and for our neutralization assays in mammalian cells. But for our yeast display campaign to affinity mature our design proteins, we did do all of that in yeast, and the glycosylation is likely to be different. So this could well be the reason why we see that our tightest binders from yeast display don't express super well in the mammalian cells. When we try to purify them, we get lower yields, and it's more difficult to test their binding. So, yeah.
Great. Dr. Jeff Glaska, would you like to add anything?
No, that pretty much sums it up. There is a difference between our engineering platform and our actual final expression platform that it's tough to take into account.
Okay, great, thank you. Dr. Silver, has there been any studies of COVID having possibly lain dormant in one's cells?
I'm not enough of a virologist, to be honest, but you know, we have the phenomena of people who carry the virus and have no symptoms. So I don't know if you call that dormant. Presumably that is, well, there's people who study this way more than I do, but that's a product of the interaction between, in part, the interaction between the virus and your innate immune system. And that's what make people, it's a fascinating human genetics problem as well as what makes people asymptomatic. We're going to learn a ton, but keeping in mind, it's that asymptomatic component of the virus that has made this such a problem and why there was such an early outbreak. I don't know, but it's not a retrovirus. It doesn't. So anyway, I don't know about that. It's an RNA-based virus, so that's how it works.
Indeed. Dr. Collins, would you see your detector potentially replacing QRT-PCR for detecting COVID?
No, I don't see it replacing it, but I think it can serve a slightly different role. I think we need multiple tests out there. I think the RT-PCR is still the gold standard. I think it is a gold standard even relative to what we have. I think what our platforms can offer are cheaper, faster alternatives that, against some standards, give similar results, but I think the RT-PCR is still going to give you much better results. The beauty of some of the things that we and many other groups are developing are the easier to use and portability of the systems compared to some of the RT-PCR efforts.
I see. Dr. Silver, you also discussed sort of a detector in your DNA living biosensor. Are there any dangers perhaps in using a bacteria as a biosensor?
Well, it depends on how, well, there's two ways to look at that. The only way to look at that question is if you were going to release it into the environment. And so, I mean, if you're going to use it as a laboratory test, for example, you have a bunch of water and you're going to add it and shake it up and get a readout, then it's being used under a genetically safe environment. Release into the environment then falls under the rubric of a genetically modified organism. And this is something that's a bigger question in how are we going to deal in the future with the benefits that we can reap from using synthetic biology tools in the environment. And that's a complicated science question as well as a political question and a sociological question. As synthetic biologists, we work very hard towards making systems that are going to be safe for the environment. And with regard to bacteria, there are ways to do that. This has huge implications also in the ag industry, where the impact of engineering can be extremely beneficial to the entire world. And so this is something we have to figure out.
I see. Thank you. Dr. Baker, can we screen small bacterial proteins against perhaps the targets identified by deep learning randomly?
We've been developing protein design methods for designing binders, high-affinity binders to targets. All require knowledge of the three-dimensional structure of the target. So as structure prediction methods become better, and there has been a lot of progress, then you could imagine taking new candidate targets and predicting their structures and designing against those. But thus far, we've really been focusing on targets for which cryo-EM or crystal structures are available, which turns out to be most human therapeutic targets of interest.
Thank you. Dr. Anam, what is the potential efficacy? I know you've done it only in non-humans, but what is the potential efficacy of using these receptor traps, such as AC2?
This is a hard question to answer without having done the experiment in people. You're asking what's the efficacy in people?
Yeah, what would you predict, or would you expect it to be as efficient?
It's a function of many different factors, right? It's going to depend on how much of it you can get into the infected tissue, and the affinity, of course, of binding, and the amount of time it spends in the blood and in the tissue. These are the types of things that we're trying to understand now in animal models. We're hopeful that it looks like from preliminary data, like there could be an advantage in using a receptor trap strategy to get the drug into the lungs. We see, it's really preliminary, but we see a higher concentration in the tissue as compared to control antibody. So that could be one advantage, but I don't really have a great answer to this question without doing more experiments and testing it in something that is in a model system.
That's fair. Dr. Jeff Glasgow, would you know of any biological risks are there any known biological risk of using a receptor trap?
Well, one concern that has come up is you're through injecting ACE2, recombinant ACE2 into a person, you run the risk of inducing an anti-ACE2 immune response, especially since ours is a slightly mutated version. You can imagine getting an immune response, and that would cause a autoimmune reaction. That's that said, there are receptor traps for other using other human receptors in the clinic now, and that hasn't been a huge issue. But it's something that we would definitely need to evaluate when we get to the human trials.
Great, thank you. Dr. Collins, how far away from the application stages are the mask technology or the wearable diagnostic technology?
Well, so they're at the application stage from an academic standpoint, in that we've now done a number of different proof of concept demonstrations. From, say, the product stage of where it will be released, I think it's probably still several months away. We have had tremendous interest from mask manufacturers and various clothing groups that want to incorporate it. We've not chosen a partner yet, and we will be publishing the works very, very soon, and expect, again, getting a lot of interest. So we're trying to figure out what's the best way we can move this out. Having said all that, I actually think we probably will just end up focusing on the insert for the mask, which would be independent of any mask that then could be made available. And that would just involve these foldable paper-based microfluidic lateral flow assay type systems that I mentioned earlier.
Great, thank you, Dr. Collins. Dr. Silver, you mentioned that these assays could be used for new therapeutics. Can you talk a little bit more about that and perhaps give an example of how?
So yeah, well, first of all, so these are gene-specific assays, and so as opposed to whole virus assays. So you can use these assays if you wanted to make a drug that targeted a specific gene in a certain pathway. You would have a rapid readout. Also, well, I use the example with NSP5, which is a protease, which is a drug targetable enzyme, and that we discovered this in vivo function for it. And so if a protease inhibitor reverses its effect on the innate immune response, then that could act as a very rapid in vivo assay Of course, in the end, all such drugs would have to be tested on whole virus, but this could be a way to bootstrap your way to interesting drugs.
I see. Thank you, Dr. Silver. Dr. Baker, do you see, you mentioned your sensors being used for vaccines, but do you see them being used for potentially other applications?
Well, let's see. Yeah, I gave an example of a troponin sensor. I mean, it's basically what we've designed are tiny molecular devices that you can basically make respond to almost anything. So it's a pretty general platform for sensing. So we've made thus far sensors for the virus, for antibodies against the virus, for a variety of cellular receptors, for antibodies, just generic antibodies. So yeah, I think that the technology itself is very general. All it requires is a high affinity binding domain to the thing you want to sense, because then basically the idea is the free energy of binding of that binding domain to the target drives opening of the switch, and that results in the emission of light.
I see. Thank you. And now we have time for just one more question, and I'm actually going to direct it to all of you. I'll start with Dr. Anum Glasgow and Dr. Jeff Glasgow. You've all spoken a little bit about how synthetic biology has advanced your research. I'm curious if you can talk just a little bit more about that. What aspects of synthetic biology has advanced the science that you have been able to put out in this time of a pandemic? Dr. Anum?
Yeah, loaded question. Yeah, I kind of grew up in synthetic biology, I guess. Like some of these guys have established the field, but for me and Jeff, we joined a lab in graduate school where like new cloning methods were coming out really fast, and it became easy to build a lot of constructs at one time and develop assays. I guess like for us, it's been about the tools as well as the way of thinking about these problems. So being able to model biological processes and predict outcomes really informs the way that we like structure projects and try to answer questions about such biological systems, and then the tools that are available and always being developed in the field and to which we try to contribute, make it easier to engineer new and interesting tools, therapeutics, diagnostics, all types of things. I don't know, Jeff, what do you think?
Yeah, for sure. The advances in DNA synthesis and cloning techniques really make the high throughput screening that we did possible.
Great, thank you. Dr. Baker, how has synthetic biology advanced your science, or where do you see it? How do you see it continue to advance science in general?
Yeah, well, I would really echo what Anum and Jeff said. A number of years ago, when we wanted to design proteins to have a new function, we would do the designs on the computer, and then we would choose our best 10 or 20 designs, and we'd make synthetic genes and then make the proteins in bacteria and see if they functioned. But now, because of advances in gene synthesis, particularly chip-based oligosynthesis, for small proteins like the binders I described, we can now design hundreds of thousands of proteins on the computer, each one completely new and having a completely unique amino acid sequence, and then get a 100,000 genes that encode those 100,000 brand new proteins, all in one tube for a relatively low price, and then we can put them into bacteria or yeast cells and evaluate each one of them. So this idea that you could be testing, you could make up 100,000 new proteins and then bring them to life by gene synthesis on this scale is just really amazing. And I think it's gene synthesis technology has really driven a lot of the progress in the field.
Wonderful, indeed. Dr. Silver?
Well, so first I want to begin by saying that it's really that one thing is the people. So seeing two younger people here and their enthusiasm. I think that's one of our greatest resources and advancements, and training people for the exciting future. So that to me is really one of the main drivers. And then I have to echo what David said. When we first laid out the roadmap for synthetic biology a million years ago, I can't even remember when, Jim will remember, it was really the faster, cheaper, more predictable design of biology was at the core, and DNA was the substrate, is the commodity for making this all happen. And so I'm just going to echo what David said, that this ability to basically screen protein libraries is, I think, just a huge breakthrough. And we do this in a couple different projects. And I think, and that's pretty novel. When I give talks about this, people see that as one of the interesting novelties that synthetic biology is bringing forward, and they say, "Oh, what a great idea to screen whole protein libraries." Albeit that had been being done in the past, but probably not at the scale, as David said, that it can be done now. So we're really at the faster, cheaper part of the equation, and now hopefully we can get better at the prediction.
Yeah, wonderfully said. Dr. Collins, would you like to add?
Yeah, I mean, a few things. So I think in the midst of the pandemic, we were able to harness synthetic biology to help us advance molecular diagnostics, from CLIA lab-based tests to point of care to at home to wearable ones. And I think it was the programmable nature of biology that's enabled by synthetic biology that allowed us and others to do that. It's curious on kind of the discovery aspect. I think it's one of the spaces for real growth in synthetic biology, that is, as a tool to enable discovery. I don't think we've had as much in the field. Most of the effort in the field has been around design and development. But I think over the next decade, we'll increasingly see folks turning to synthetic biology for discovery, whether it's kind of the patch clamp equivalent in neuroscience, but now molecular biology, and or as developing new and interesting screening tools and capabilities that will expand our ability to discover. And let me just close that. I think as I look out, I do think synthetic biology, broadly this idea of engineering biology, is going to be one of the defining technologies of this century. And we're just beginning. Biology is far from being an engineering discipline. And I think it's really exciting for young people in particular, with many, many decades ahead of themselves for professional work, to get into this space and use biology to address some of the big problems we all face in this world.
Wonderfully said. It does seem to have certainly come of age. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our panel of speakers and Q&A participants, Dr. Anum Glasgow, Dr. David Baker, Dr. Jim Collins, and Dr. Jeff Glasgow, and Dr. Pamela Silver, as well as our sponsors, Beckmann Coulter Life Sciences, Isoplexis, Twist Bioscience, and Acro Biosystems. Thank you, everyone, and have a great rest of your day. Goodbye.